I'm Kula Lifshitz, and it's now my pleasure to introduce our next guest from VibleLogic, Tom Hockley, Chief Executive Officer, Dr. Kai Lipinski, Chief Scientific Officer, and Joe Sinclair, Vice President of Business Development and Corporate Strategy. So the format for this session is a 30 to 40-minute fireside chat. And if any of our audience attendees have questions during the session, feel free to type them into the question box under the Zoom window, and we'll try to ask them on your behalf. So thank you all for joining us today. And to start off, I'll ask the speakers to briefly introduce themselves, their role within VibleLogic, and any background to share for the audience.
Well, why don't I start? I'm Tom Hockley. I'm the Chief Executive Officer of VibleLogic. I joined VibleLogic in July of last year after spending a lifetime leading operations for pharmaceuticals, biologics, medical device, and human tissue. Started my career, first 10 years at Johnson & Johnson, also spent a decade at Integra Life Sciences. And lastly, in the last 2 years prior to coming to VibleLogic, I ran the site for Houston, for Lonza in Houston for cell and gene therapy. Kai?
Yeah, hello, Gil, and everybody else on the fireside. So I'm Kai Lipinski. I'm the Chief Scientific Officer at VibleLogic since April last year. And before that, I was head of process development for nearly 10 years, also for VibleLogic. By training, I'm a molecular biologist with a PhD in transcription regulation. After my PhD, I worked for a UK-based CDMO, Cobra Biologics, in various roles, focusing at the beginning on IP generation for cancer gene therapy products, and then later on for process development and tech process transfer to GMP. So as CSO at VibleLogic, I am involved and responsible for strategical and technological directions. I serve the company as internal and external subject matter expert, particularly for cell culture and virus production. And I support the BD team as SME for the acquisition of new clients and projects, and I represent the company scientifically.
Thanks, Kai, and good afternoon, good evening, everyone. My name is Joe Sinclair, Vice President of Business Development and Corporate Strategy at VibleLogic. I had joined this past year as a part of the company's global leadership expansion, and before working as the head of sales with WuXi Advanced Therapies, my career has been spent in CDMO, partnering with clientele, working to develop both proteins, cell and gene therapies, and vaccine products. Responsible here for corporate development initiatives and strategic elements of the business, including establishment of key and anchor client strategy and business partnerships.
Great. So I understand VibleLogic has been established since the early 2000s in the viral CDMO market. So Tom, you joined shortly after an acquisition of the company by Ampersand Capital Partners in 2019. So can you provide some insight into that acquisition and some further background on the company, as well as its future direction for those new to the story?
Sure, I'll actually start back at the beginning of VibleLogic. It was 2002, up in here in northern Germany, where Kai and I are currently this evening. It's an area known for poultry and egg-based production, and the company started on veterinary products through egg and poultry type of applications. In 2006, the direction of the company changed, and we headed more towards human type products, and that's when we changed the name to VibleLogic, which actually stands for viruses, bacteria, and biologics, where we were focusing. For the most part, in the early phases, we were a CDMO for preclinical and early stage clinical products, oncolytic viruses, viral vector vaccines, and viral vaccines. Over the course of time, the company was quite successful meeting our promises. And by 2017, 2018, company realized to go to the next level, they needed some further capital investment. And that's when Ampersand became interested in the company. Ampersand had been very successful with the Brammer sale to Thermo Fisher, and looked at us as potentially something very significant and very similar. So the company purchased VibleLogic outright in 2019. And in 2020, I joined to take the company to the next step. So again, primarily we've been early stage clinical, and Ampersand realized to expand the business, we wanted to be able to offer a full portfolio of products from preclinical through early stage clinical to late stage commercial as well. So we looked at the market and realized that to expand the business, we really wanted to have a global footprint. And I came on to establish a headquarters here in the United States, and also to build out a commercial manufacturing on the East Coast. And I'll talk about a little bit about that a little bit more in the future. But again, growing considerably here in Germany and in the United States. And we will provide a full portfolio of products to our customers.
Great, great. And so Kai, maybe you can you characterize for us where VibaLogics would be positioned overall within the context of the broader CDMO market, and maybe talk a little bit further about the company's expertise in production and analytics.
Yeah, happy to do so in much more detail now. We are really occupying a certain product niche, and that's live virus products. And this comprises really three pillars. It's oncolytic viruses, viral vector prophylactic vaccines, and viral vector gene therapy products. So we regard ourselves for these kind of products really as one-stop shop experts. And we do that, we are that in the areas of process development, GMP manufacture of seed materials, drug substance and drug product, and analytics, and at the end QC release. We also have, for example, a QP, what you call a pharmaceutical person on site, and she's 100% a VibaLogics employee. As Thomas already mentioned, Cuxhaven serves mainly the clinical phase 1-2. And for Boxborough, we will focus on phase 3 and commercial supply. As I said, we also do drug product here in Cuxhaven, and we offer up to 30,000 glass vials with automatic fill in a wrap system. And Boxborough will do the same. They will offer up to 50,000 glass vials with an isolator technology, and also with an option to have syringes filled. In Cuxhaven, additionally, we can offer lyophilization services in process development and in GMP. Here we have currently up to 3,000 vials. So more or less currently smaller batches lyophilized drug product, but we are already looking into potential expansion of that capacity also here in Cuxhaven. So with regard to the platforms that we are experienced with, we are more or less experienced with all the substrates which are known to people in the art. So we produce viruses with diploid and continuous cell lines. And we belong to few CDMOs in the world that are also very experienced with virus production in eggs or on chicken embryonic fibroblasts in roller bottles or cell stacks. And we cover the field of both adherent and suspension cell culture. If we talk about suspension, it's in Cuxhaven currently 200 liter scale, and we are currently building up a new production line for 500 liter. And as Tom already mentioned, in Boxborough, we will go to 2000 liter. Our adherent cell culture experience is mainly on, as I said, cell stacks, roller bottles, and we also have experience with pulse iCELLis fixed bed bioreactor systems. And we are also very proud, I must say, of our many years and operator experience in aseptic processing, because many of the viruses that we produce cannot be sterile filtered. So we have to perform aseptic processing and also aseptic validation. With regard to the products we are working with, it really resembles the current clinical design, and that covers adenoviruses, rhabdoviruses, herpes viruses, diverse pox viruses, and also, as, for example, AAV as an example as a gene therapy vector. So in analytics, it's a nice combination between in-house capacities that mainly covers product-specific identities, potency assays, process residuals, and physicochemical assays. And we also work with CROs for most of the safety analytical release testing at the end. Live virus also needs very specific and experienced work for titration. And so that is the main potency assay for live viruses, and we regard ourselves as experts in doing so. So we are used to all different kind and of formats of live titration that is like TCID50, plug assay, or even EGG-ID50, or different other forms which include like immunocytochemistry and immunofluorescence focus forming unit assays. So we cover basically everything here. And we also are used to perform assay validation and stability studies, all this in line with the ICH guidelines, basically.
Great. Well, thanks for that great overview. And so, Tom, you mentioned the company's growth plans and ongoing facility expansions. We see a lot of activity within the space on GMP facility construction, both on the vendor side and the development side. So from your perspective, what makes for a successful physical expansion? And can you share some of, let's say, the general insight or discuss the past experience here for the audience? And are there any unique considerations or opportunities specifically in today's environment?
Absolutely. I mean, I can go through a lot of our decision-making process for our facility, but quite honestly, first and foremost, it was the availability of talent. So as we look to where we're going to expand our business, we looked at where is the core competencies for cell and gene therapy in the employee market. So we looked up and down the East Coast from areas such as Raleigh, North Carolina, and Maryland, and Philadelphia. And quite honestly, we wound up in Boston because it is the hub for cell and gene therapy R&D, and we knew that there would be great access to employees there. So what we did is we retained a real estate company to find buildings that met our criteria. And there's a lot of variables and a lot of decision making in this process, whether you go with a greenfield or a brownfield. But what we wanted to do was we wanted to minimize the time until we got to GMP ready and minimize the risk. So we chose a brownfield facility on some strict criteria to include existing power and sewer and water. We also thought it was very important to have expansion space, most important, so that we could meet our customers' needs today, tomorrow, and in the future. So the ability to expand was very important as well. So we found a building that could easily handle what we needed today, tomorrow, and in the future. And also, again, we minimized our risk and our speed to completion by purchasing modular clean rooms from a company down in Florida called Germ-Free that we have partnered with. So as we prepare the facility, they are preparing our clean rooms, and in June, they will be brought up the Interstate 95 to our facility and moved into place very, very quickly. So again, it's the selection criteria, and by doing this now in the modular form, as just a small portion of the facility, we're learning what it's going to take for our phase 2, phase 3, and phase 4, so that we can readily expand based upon what the customer's needs are. So again, it's minimizing risk and reducing time to completion that we thought was most important to us.
Right. That makes sense. And so maybe I'll open this next question up to any of you. So from your perspective, with regards to vendor-client collaboration, what are some of the key elements of a strong partnership? And how do those components of communication and experience play into a successful engagement with a client?
So to be happy to take that question. And for me, seeing many relationships over the years, there are really a few key components which always seem to be a part of that experience, and communication in particular usually is aligned to all of them. So one for me is fit for the product and the process technology itself. The second would be really around project planning and execution of activities. And the third being around partnership or more of those longer range projection activities. For the first one, fit for product and process, due diligence is really important here at the end of the day. I think there are a lot of standards in the way that companies are approaching this, but you really need to understand both the technology and the modality around that being fit for partnership. That's looking at both direct experience of that CDMO partner, and if not direct, indirect experience should also be looked at and how that could potentially apply. From the client side, the relationship evaluation traditional through an RFI or RFP process, site or virtual audits and visits, but most importantly, or importantly as well, the early sales, technical and quality discussions, and bringing parties together with those who are really going to be hands-on with the project. Making sure depth of discussion occurs there is really going to be important. I think in terms of the second aspect here on project planning and execution, we're working in an incredibly dynamic environment right now. And from the client side, pipeline priority adjustments are common, clinical phase delays, even failures potentially, or pressures from those influential parties, potentially third-party investors, can help shift and put pressure on the direction a project is going. And the vendor side as a CDMO, also very complex. We're managing materials procurement, we're mapping project schedules to available capacities, we're executing to the project ultimately, and even potentially hiring up employees or FTEs in support of that collaboration. Program delays and unforeseen events come up from time to time as science and biology don't always work the way we intended to, but where possible, fully evaluate those activities. And lastly here, Gwella, around partnership and longer range planning, really staying ahead of activities through good communication there. Looking 6, 12, 18, 24 months out is extremely important when it can be done. I know that's often or occasionally a challenge to project and forecast, but this will allow partners to really remain in position to deliver the best results around a project. And last, having a good structure around that communication. It could be setting up periodic joint steering committee meetings, or at least making sure there's a mechanism to support the relationship at the most senior level of the company, and opening that up for discussion there really can have meaningful impacts for both parties, both client and vendor at the end of the day.
Mm-hmm. Great. And so maybe just a quick follow-up. So from your perspective, in terms of risk benefit, are there certain tasks that the emerging biotech company can take on versus ones that are best left to the CDMO to perform? Or is this really case by case from your experience?
Kai, do you want to run with that question, maybe from a technology and process transfer standpoint?
Yeah, I mean, absolutely. There are always certain things the product owner should certainly take care of. I think for a newcomer biotech company, it's really important to put together also at the beginning a strong team. So I think the academic people, they clearly need support from the industry and consultation leader. It just needs more than only science to make really a successful biotech product company. And yeah, sure. I mean, even the product developers, they can do some early on product and development and characterization just to show proof of concept for their product, and also maybe the process, the early lab scale process. And so they could do already some work, everything that's not on the critical GMP path. And even occasionally, the product owners, they do some activities on the GMP path. We as a CDMO can only recommend and watch your material compliance really. So take care that you have not a bad surprise later on when you have to reveal what materials and raw materials you have used if you have done something on the GMP path. So that could be the truth for like FBS or any hydrolysates or hormones like insulin. Now albumin is becoming more and more important as an ingredient for production and also for formulations, or any other ingredients. And more we see now recombinant substitutes for animal-derived components. So have a look out for these kind of products if you really think you want to do something that's on the critical GMP path. Again, early on productivity, the product owner can have a look at. So have a sanity check here. So do you have a viable product for the market? Is the productivity, or do you think you get the productivity to a point that it will be a viable product? That's also important. And we always tell our clients, try to talk to the regulatory agencies early enough to make sure your QC assay panel, your animal models, and your clinical trial design is on solid elements and that it all makes sense and comes together. That is what we tell our potential clients. And most product developers also will early on look for potential collaborations and maybe also define the exit strategy, because going through phase 1 to 3 clinical development is very expensive. And there is hardly any biotech company that can stem themselves, and at some certain stage, they probably, beside private equity and other investments, they will look for a larger pharmaceutical company partner that will take them along through the later clinical phases. So yes, there's a lot of homework that the product developer can also do, and we are just coming in a little bit later normally. Yeah.
Great. And so, as has been discussed over the course of today, biotech companies often face the decision of developing internal manufacturing versus working with a CDMO partner. So from your perspective, what do you see as the core advantages of working with a CDMO for a smaller biotech versus deciding to invest in internal?
Well, let me answer that question. As Kai just mentioned, there are a lot of risks and costs associated with drug development. And I think by partnering with a CDMO, it reduces that risk and also that capital investment by large companies or small biotech as well. Because we already have the infrastructure, we have the brick and mortar, we have trained workforce, and we have methods that we've used in the past that have been successful. Thus reducing the risk, reducing the capital investment, and also aiding in getting that product to market quickly, because we have readily available capacity and services that otherwise the company would probably have to develop. So we pretty much streamline the process and reduce that risk, and that's probably one of the biggest benefits.
Got it. Right. And so you mentioned earlier a number of capabilities across different viral modalities. So as customers approach your business, what are some of the core product technologies or modalities that you're seeing approaching? And are you noticing any trends that are forming or a shift in terms of trends from prior years?
I'd be happy to take that. So I think the interesting thing here is the modalities are following both the science as well as the investment trends and investment potential. Commercial approval in gene-mediated cell therapies, and including CAR T products, TCRs, and some of the in vivo gene therapies has really accelerated the market activity there, where a majority of funding remains flowing into also immuno-oncology and is creating a tremendous amount of activity. Vibalogics, and due to our unique position, we're seeing particular demand actually in oncolytic virus programs. This is still an emerging market, only about 150 or so ongoing clinical trials, but picking up good momentum, good funding. Much of the market there is in early phase clinical, and really due to Amgen's commercial approval of Imlygic, they really created a regulatory pathway or a template to follow for many of these companies subsequently. And the activity has certainly been picking up over the years due to ultimately confidence there. Within our business, we're seeing a broad range of viruses and vectors, and looking into that oncolytic class, about 30 or 40% of market right now we see in the adenovirus space. There's another subset in herpes simplex virus. We're seeing what seems to be an acceleration of vaccinia or modified vaccinia programs, also some others such as VSV and more. Each virus, of course, has pros and cons when it comes to application and packaging potential of that virus. or its wild type properties, as well as safety considerations such as immunogenicity, so on and so forth. So that's what we're seeing right now and representative of the business coming to VibaLogics.
Got it. And then within that oncolytic virus space, looking ahead, do you envision the field continuing in the same breakdown or other trends that you might expect moving forward?
Kai, could I have you take that and
Oh,
further
yeah.
follow up there?
Yeah, happy to take that on. I think the next maybe 5 years will be very important to the field. So we're having a few more products that will reach late phase 2 and 3. And after the approval of IMNIGIC, I think the scientific scene is, and the OV scene is particularly waiting and looking for another market approval of another product. So that's very critical. And I think the next coming years, we will probably find out that some OVs work clearly better than the others. So some natural selection of a fewer platforms to survive probably. And this is also because the oncolytic virus inherent immunological features are quite diverse. So this will also affect the anti-tumor immunity and efficacy in the clinical phases. And I think we will see that some OV platforms work better for systemic compared to others. And for some, you maybe just have to go for intratumoral administration. As an example, vaccinia virus currently, pox viruses are the lead candidates maybe for systemic application. Yeah, besides the most commonly used OV platforms, like we named them already, herpes viruses, adenoviruses, pox viruses, we also have a continuously feed of very new platform types. And hopefully, we will be very positively surprised by at least some of these newcomers. So yeah, maybe a few examples. We have vesicular stomatitis virus. We have 2 companies here, at least, who are developing those. We have Viriat in the USA, and we have Vera Therapeutics, which was taken over by Boehringer a couple of years ago. Then we have arena viruses, and here we have companies like HOOKIPA and Abalos. Again, Abalos. Here we have Boehringer Ingelheim involvement. Another company like Myxoma virus, Oncomyx, again, a company that is connected to Boehringer Ingelheim. We have alpha viruses, another platform. Example here is Venezuelan equine encephalitis virus. So many new platforms, and I'm sure some of them will generate very interesting preclinical and clinical data. Another focus is the cargo genes, which we use to load the oncolytic viruses. And I'm sure here also we will find out which cargo genes work better than others, and which combination therapies work particularly well compared to others. And here we can choose from, besides the oncolytic viruses and the cargo genes, we have the immune checkpoint inhibitors, we have chemotherapy, we have radiation therapy, and also let's say the targeted CAR T therapy or other tumor infiltrating cell therapy. And so the number of combinations that we can imagine is very high. And here we have to find out what works best really. Other technologies, for example, we have seen companies that develop shielding technologies to prevent the immune system from recognizing the viral vector. There's a company, for example, Valo Therapeutics. They have a peptide shielding technology called PeptiCrut. It's also very interesting. And additionally, I think what we call the platform processes are very important because they serve to go quicker through the process development pipeline and use one platform process potentially for more than one kind of OV viral platform. I think what we will also see is more and more collaborations between large pharma and biotech, because We have here a nice complementation of very different products. So we have the immune checkpoint inhibitory antibody, we have the OVs, and we have the CAR-Ts. So with these takeovers, we see more and more clinical trials with these combination therapies. We will see also a lot of improvement, I think, for processing with regard to analytics and processing analytical tools. Nice example here is a digital droplet PCR, which will probably in a very few years will have already substituted the standard quantitative PCR. And we see new technologies emerging, particularly for the analysis of viral particles. So we have what we know flow cytometry is now also coming up for viral analytics, or we have even electron microscopy in a smaller formats available, which we might find in a few years in CRO companies also. So a lot is going on here, yeah.
Great, well, that makes sense. So maybe as we think about commercialization, I'd like to talk about scalability. This is something that's been touched on over the course of the day as well. So scalability and reproducible processes for vectors at larger volumes or for larger disease states have been one of the rate limiting factors in delivering treatment to patients. So from your perspective, what are some of the challenges and opportunities that you see here? Can platform processes help better manage these issues?
Yeah, everybody's talking about scale, certainly an issue, but there will be a limit somewhere. Currently, we are about at the 2,000 liter scale, but we know that Thermo, for example, also offers now the 5,000 liter, the first 5,000 liter single-use bioreactor. As a CDMO, we clearly see a trend towards suspension processes because they are much more easy to scale up, but it's not always possible. Sometimes if you change from adherent to suspension, you reduce the productivity. That's sometimes to allow the developing company, the product owner, to divert to a suspension process. So for some viruses, because they are dependent on certain cell lines, you still have to stick to the adherent cultivation of cells. And here particularly, we have the scaling issue. We have, for example, the HyperStacks from Corning. They have become one of the workhorses really in the industry. It's one of the best systems you can have when you are still at, let's say, small to mid scale. But for very large scale, again, it's very difficult to scale it up really. And here we are currently left with maybe two opportunities, and these are the fixed bed bioreactors. And we have one available from PAL, it's the iSystem. And recently, a new company, Unilever Cells, come up with a new generation of little bit comparable iCELLis system also in some respects. It's a ScaleX platform. And this is also very interesting. So with these two fixed bed bioreactor systems, we now have something at hand to scale up the processes really to a reasonable commercial scale, I would say. And we also know that the number of cell lines you can choose from, they are restricted. So we have cell lines, they are adaptable very well to suspension, like HEK cells, we have HeLa, we have MDCK, and we have a few proprietary cell lines, like from Johnson & Johnson, the Percy-6 cell line, or from CEVEC, the CAP cells. But then we have other cell lines we adapt to suspension. That is the Vero cells, A549, or some diploid cell lines, like MRC5 and WI38. And also the primary egg-derived CEFs you cannot easily adapt to suspension. Yeah, coming back to the platforms, platform processes are key to give CDMOs a selling and a competitive advantage, certainly. And I'm sure that all CDMOs are trying to establish platform processes. They really accelerate your development timelines and they reduce your cost. And they give you a more robust process at the end because you can perform them again and again, more or less, because it's the same platform. What we also see is, on the other hand, that the productivities are moved We see so many new and great media chemically defined coming along and new supplements. And they serve to increase productivity and thereby potentially keeps the scale at a reasonable maximum scale. Then we have the producer cell lines. They make it easier to publish platform processes, and hopefully also they will be able to come up with higher productivities. I also like the idea of, let's call them intelligent and designed cell lines. For example, you can knock out in cells certain genes which downregulate virus production. It's a very natural process that after a cell is infected with a virus, downregulate normally the ability of its own machine, its own cell environment to that the virus cannot replicate itself. For example, ATCC is offering a cell line, it's called Vero STAT1 knockout. So STAT1 is a very important pathway in viral regulation. And such a cell line has been designed with intention to increase viral productivity clearly. And I'm sure we will see more types of this kind of designed cell lines coming along potentially. Other important developments are perfusion processes because they allow very high cell density cultivations, and that directly pays out higher viral productivity. And then as a good compromise between adherent and bioreactor culture, we have the microcarrier cultivation. So here you have a cell line that is storage dependent. They need the microcarrier, but you can still cultivate them in a scalable bioreactor environment. So yeah, so I'm very keen to see the next 5 years, what's gonna happen. And as I said at the beginning, I'm saying there will be some consolidation in all areas that we are currently looking at. And I'm very optimistic that in the next 5 years, we will have at least one more market approved OV, I would think.
Great. Well, you just touched on a lot of great innovations within the manufacturing space that I think was going to answer my next question, which was in terms of these technologies, how they're likely to shape the manufacturing landscape in the near term versus, let's say, 5 years from now. So within any of those, any brief comment on how you envision that playing out, or is this one of those scenarios where we'll see how the space evolves?
Kai, maybe I'll take some of this more from a nuts and bolts or market perspective, and you can follow on with any technology improvements or advancements there. I think from my perspective, looking at this from a market capacity standpoint, if you put that in grand context, for genetic therapies, manufacturing capacity is still only about 5% of global GMP production. So the initial focus here, both through drug development companies and CDMOs alike, will be on just sheer volumetric production. Just build out the rooms, build out bioreactors or the technologies to scale there. We've seen recently an incredible amount of both buy and build activity, as well as M&A in the space. Single-use manufacturing is also another interesting component. There have been a lot of advances there recently. Modular facilities, such as the ones that Vibalogics is engaged with, are making it ultimately easier for companies to build and scale quicker and be more flexible in the process. So things such as prefabricated clean rooms, they can be delivered globally, if you think about this, to different production centers where those would be set up, and make that GMP manufacturing more accessible as well around the world. As Kai had also shared, a number of the technological advancements would certainly be needed by way of process. Companies and CDMOs alike are focusing more, as he stated, on platform development. That could be components of platform cell lines, media and formulations, your upstream and downstream processes. Ultimately, at the end of the day, that will save you both time and cost through having a more reproducible and better understood process. And you'll also get quicker throughput and better performance predictability at the end of the day. Kai, anything else to add from your end more from a
Yeah.
technology standpoint?
I mean, as I mentioned already, we are hitting scale maximum currently at about 2,000 liter per production batch, and this is part is due to the limitation of in the cell substrates themselves. And but as I said, many companies are working on robust production cell lines, which can much better align to the volumetric equipment and vessels that we see currently. And as I said already, that we now see the first 5,000 liter SOB. So let's see how the market's acceptance is for this kind of single-use bioreactor scale. Yeah. With regard to productivities and titters, I mean, it's very dependent on the products also. So for some products, we may be hitting 10,000 doses we can produce per batch, but we have some, particularly the gene therapy products, where in the worst case, it comes down to one dose from one complete clinical batch. So here we really have to drive down the production costs, and we have to increase the dose supply per batch. So I'm sure the industry and also the CDMOs will remain very focused and try everything to advance here the technologies which are available. A bias still is the usage of, let's say, animal-derived components like FBS for adherent cell culture. And again, many companies, suppliers here define new and better chemically and animal protein-free media, and they can be very good. And so hopefully this will come around much more so that we can in the end get rid of most of the production systems that contain FBS, for example. So transient transfection is still a burden on the industry to produce particularly lentiviral and AAV. Plasmid demand is very high, and it's also adds a lot of costs to the overall projects. And again, here, the solution might be in the future for many products to use and generate stable production cell lines. So we as CDMOs are still approached sometimes early on from academic labs. And the processes that we transfer to our facilities are often still at very early academic grade, and there's a clear need to mature them to something that is adaptable to GMP. But that is also a good thing for us, obviously. And in the end, new technology platforms arising here, like what we have seen now for COVID, mRNA vaccines. So they are also very interesting because they decrease the timelines you get until the point that you have produced your batch, and they can also be produced likely with reduced cost compared to the, let's say, attenuated viral vaccine, for example. So, yeah.
Yeah, definitely lots of learnings that can be gleaned from the rapid advancements of the COVID vaccine space. Well, fantastic. I think we're just about at the end of our session. So I really appreciate you taking me through the various technological applications that VibaLogics is advancing, as well as all of the technological innovation that we can anticipate within the manufacturing space. So I want to thank you all again for joining us.
It was a pleasure.
Thank you to Chardon, and thank you, Gwilda.
Thank you.